thanks. Well, yeah, indeed. So I think discovering of de novo motifs is one of those classic problems in bioinformatics. It's been around for more than 20 years, and solutions go back to the 90s, and I will show one of them. And then still we're missing good algorithms to apply to the data. And just to give an outline before I start, so I'm going to first cover one of the classic algorithms, the MEME algorithm, and it will be a reminder, I guess, for maybe some or most of you, if you know the algorithm, and also about expectation maximization. Then in the second part, I'm going to move to and discuss new technologies and the kind of data that they provide us, and also the computational problems and solutions that arise from these new technologies. And if I go too fast through the the first two parts, then I also have kept some slides at the end about sequence designs problem that arise from these technologies and are totally theoretical. So maybe we'll be able to discuss those. If not, then I'll discuss those in the workshop on gene regulation. So I'll start with the first part. And I just wanted to say, so these parts are independent. So if you have a learning breakdown in one of those, then you can still get the other part. Or if you fall asleep and it's after lunch and you already know the material. So let's start with de novo motif discovery using expectation maximization. And I think it will be a good reminder because EM is a great algorithm. It's very useful, and many bioinformatic problems use it. And the slides are taken from the Computational Genomics class at Tel Aviv University, given by Professor Oded Sharan and Ron Shamir. So I don't think I have to go over this slide because Nadav gave an introduction and motivation of 1.5 hours about why we want to be able to identify the binding sites where the protein bind and control gene expression, and want to model those so we can predict new binding sites and understand the regulatory mechanism behind transcription factor binding. And I think the story that complicates things about the binding of protein to DNA is that it does not bind to a single sequence. It may bind to several different sequences. So here, for example, we have the protein and the DNA molecule, and we see that the protein prefers to bind to this sequence, ACGT-GCA, and let's say it has some intensity, 100 in some units. But if we make a mutation in that sequence, or if it appears in the genome with some mutation, it may still bind it, but with a smaller intensity. So both of these can serve as binding sites, but one is stronger than the other in terms of binding intensity. We can also make a different mutation in the same position, and we will get a different intensity. So there are different potential binding sites or sequences that serve as the binding site. And it can be also that we make two mutations, and then we lose any binding, and the intensity is the background binding. So what we would like is to model these, to have a model that will be able to give us an ability to predict the binding site and intensity to the genomic sequences or DNA sequences in general. So what are the computational models that are common and used to represent the binding preferences? And the word motif is used to describe the set of binding site sequences that together constitute this motif. So the first and most simple model would be a consensus string. So we can just say, okay, this is the string that the protein binds, and that's it. But we've seen that this is very limited because in reality it does bind different sequences with mutation and variations. So we can relax the definition and allow it to have up to D mismatches. So in this example, we can say that we allow up to one mismatch, and then we have different binding site. But if you remember the example I just showed in the previous slide, it could be that some mismatches are allowed and the protein would still bind, but if we have a mismatch in a different position, it may not bind. For example, it may be that the first G is very important for the binding of the protein. So we must have a G in that position, and allowing any mismatch, a mismatch at any position, does not follow these rules. So we can extend the model of the consensus string with up to D mismatches into a model that we call a degenerate string. So here we are using characters, and each character does not represent a single nucleotide, but a subset of the nucleotides ACGT. These are called UPAC characters. And for example, here we have a W, which represents A or T, and B, which represents C, G, or T. So we can see these kind of variations in the binding site, but we do insist on having GG and AT in those positions. Then when we look at the sequences, we can identify those binding sites. But still, this model is also limited. And for example, in the example I've shown, one variant reduced the binding to 80, while another variant at the same position reduced the binding to intensity of 50. And this cannot be modeled using a degenerate string because we assume that they distribute uniformly, that these 80 options distribute uniformly and appear with 50% for each nucleotide. So we can extend this model into what is called the position-specific scoring matrix or position weight matrix. It has a different name. And this is the most common and used model currently and used in any tool that does prediction of new binding sites. So what we have in this matrix, we have 4 rows, each corresponding to a different nucleotides, and the number of columns corresponding to the position in the binding sites. In each one of those columns, we have weights for each of the nucleotides. This can be, for example, in this illustration, the probability of observing that nucleotide in that position. So for example, in the first position, we usually see T with 90% chance, and we see A with 10% probability, and then we never observe a C or a G. So this is the example given here, and we can also get a score for each binding site. So we can go over the 6 mers in this sequence. We can multiply the probabilities and get a score and say that if the binding site is over the threshold, in this example 0.009, it is considered a binding site. And this is a very useful model, and I think that there is a phrase that says that all models are wrong, but some are useful, so this is one of those useful models. And it has been shown to be an untrue model, and what is wrong about this model is that there is an inherent property of independence between the columns. So if we have a contribution from this column, it is completely independent from the contribution of observing a nucleotide in the next column. And it is known that there is dependency between position in protein DNA binding site. But still, it is very useful. And the cases where we do have these dependencies are very few, at least from what we know from the current data that we have. So for most protein, it works very well. And still, it's been used and still being used in all of the bioinformatic pipelines. And one of the reasons why the model is so successful, and I think I heard Gary Storm was saying that in ISMB in Berlin in 2013, that one of the things that pushed the PWM to be so popular, it's the interpretable visualization that accompanies each matrix. So we have a way to visualize the preferences in each position using the sequence logo, as it is called. And here we have the distribution given for each position. So for example, here we have 50-50 for C and G, so we have half of the occupancy of the height given to C and half given to G. And what is the height of each character? That's the opposite, the inverse of the entropy. So as the position gets more strict, the letter is higher. For example, in the first position, we have 90% given to T, so we see that this is a very high character. In a more uniform position, we see that the letter is very low. So that way we can get a sense of how strict it is positioning. And the exact calculation, would be of that height. It's called the information content, is the sum, or is actually subtracting the entropy from 2. So that's log pi. All right, summing over A, C, G, and T. So that's the height of each position. So that's the PWM. And now moving to more extensive models. So for example, the K-mer model that makes no assumption. So we can model any dependency between position. We just have all possible K-mers. And for each K-mer, we have a binding score. So that way we can express whatever dependency there is in the data. So what is the limitation in the model? It's the number of parameters. So you can imagine that if the length of the model is k, we'll need 4 to the k parameters. As opposed to this model, where in each column we have 3 parameters. If it is a distribution of 4 parameters, so we need 3K or 4K parameters. So this is a much more compact model. And when we have this, and this is exponentially greater than the PWM, which has the risk of overfitting when we don't have enough samples in the data. So that's why the PWM still has a lot of success compared to the Kramer model. In addition, what's missing for the Kramer model is that interpreter visualization, right? In the end, you want to sell your model, you want to say something that you can look at and understand what's going on. When you have a list of Kramers and scores, it's very hard to understand what's going on behind the TF binding. And there are other models that are in between the position weight matrix and the Kramer model. So supplements the PWM in the form of dinucleotides contribution. So most of the dependencies that have been observed in protein DNA binding are between 2 positions and mostly adjacent positions. So we can just add features that cover those dependencies. Other suggestions are using DNA shape features. So this is something that coming from Remo Ross lab in USC. And these features tell us how the DNA base pairs are standing one next to the other. And they also have an effect on DNA binding. Okay, so this covers the model, and what I'm going to show in the next slides and in the presentation, I'm going to focus on this model and algorithms that learn the PWM model to represent protein DNA binding.
Can you speak to how much information is there in the Kramer models in biological sense that is captured that's not in the PWM? I can like have a measurement in AUC, for example, like how well they improve the prediction of binding. At the time the models were developed, people didn't know, and now there is data to begin to answer that.
Yeah. I mean, new data, which is high throughput, allows you to infer these Kramer models. So I mean, it improves significantly. So maybe the average AUC for a Kramer model would be 93. For the PWM would be 88, something like that. So I mean, I don't have the numbers, but it's something in those.
Half the error, roughly, you're saying.
Sorry?
That's roughly halving the error.
Yeah, I mean, I think they are definitely more accurate when you have good training data. So that's a problem. So I'm talking specifically about PBM and high-throughput SELEX, which I'm going to talk about later. So there, the Kramer model is very successful. But when we now take the Kramer model and apply it to in vivo data, we don't see the same improvement. And actually, the PWM performs better. So I mean, and taking in vitro models and predicting in vivo binding is a big problem in the field. Yeah. So I'll discuss it a little bit later. Yeah.
Isn't there also an issue about the size of K? Because
yeah. So in the past, most of these models were about 6, 8, 10 base pairs, not longer. But with more data, we do observe an effect of the side position and flanking positions in addition to the core positions. So the technologies are improving and allowing us to measure the effect of the side position. But yeah, we would like them to be as long as possible. But so currently, yeah, it's still an open question. How long do we need the models to be? Yeah, but we indeed the classic or the dogma in the past that it's only 6 or 8 base pairs is no longer valid. Yeah. Okay, so if we are given a set of binding sites, then creating the PWM, the motif profile, is very easy, right? We have the set. We can just look at each position and construct the PWM according to the frequencies. So we can say this is a probability of a 3 over 5, and this is 2 over 5, 60% and 40%. And we can also get the consensus very easily by looking at the nucleotide that has the highest frequency at each position. But we are rarely given the set of binding sites. So if you heard Nadav this morning, he talked about different technologies that measure the binding of the protein to the DNA. They give us maybe 300 base pairs or 200 base pairs of where the protein is binding, but we don't know the exact binding sites. So this easy task of taking binding sites and making a profile from them is rarely the case. And we need more sophisticated algorithm to handle long sequences. So this is what motif discovery is usually like for protein DNA binding and for discovering new binding site. So first, what we need to identify is what is the set of correlated genes or the set of sequences in which we want to find the motif. These can be given from gene expression profiles. Here it's kind of obsolete. It says microarrays, but of course these days we can use RNA-seq to measure gene expression. And we can identify and cluster those genes that have similar expression profiles. Since they have similar expression profiles, we may assume that they are regulated by the same protein. The same transcription factors is binding the promoter regions, and so we can look for a motif in the promoter sequences. Another way is to have the location of binding using chip technology. So here it says chip, and indeed in the past we would measure the binding using a microarray that has a design of the promoters. These days we can use chip-seq, like Lior mentioned, and also Nadav today. So the chip-seq gives us the occupancy of the binding in the genome in vivo in resolution of about 300 base pairs. Now I want to identify the short motif in that set of sequences. And we can also take functional groups and assume that they are regulated by the same transcription factors. Then we take the sequences, and what is left is for us, the computational biologist, to develop a motif discovery algorithm and to get these nice sequence logos and PWMs that represent the binding preferences of the protein. So this is the problem that I'm going to show solutions for in the next slides. Now, what makes this problem such a big challenge and so difficult? So first, binding sites are short and degenerate, or maybe they are long, it depends, but they're usually between 10 to 20 base pairs, and they have degeneracy. They are not fixed. It's not a single sequence, the consensus sequence. They have a lot of variation in them. So we need to find a motif with those variations. Second, the promoters that they're usually in are long and complex. So in the promoters themselves, the distribution of the nucleotides outside of the binding site, it's not a uniform distribution. It's not even a fixed distribution among all the other nucleotides and position in the promoter. The promoters may have other elements and other repetitive regions which can also look like a motif. So this is another challenge. In addition, the search space is huge. So for example, if you want to consider all sequences, all motifs of length L, we need to consider 4 to the L sequences, and we might want to include all mismatches up to D mismatches, so we'll have to multiply it by L over D. And this is the space of motifs that we want to consider. So just for that, it's becoming quite huge, exponential in L, in the length of the motif, and also the number of sequences that we take in. And the promoter sequences that we look the motif in is also a big number. It could be thousands of promoters or chip-seq peaks of length 300 base pairs. And the data is noisy. So anything that we get from in vivo, from the cell, it has noise in it and it's not accurate. So that can also mislead our motif finding algorithm. So here is an example of what is exactly motif finding given these sequences. So if we implant a motif, let's say this stretch of A's and stretch of G's, it is quite visible given the rectangles, right, that go around it. But now, is it still visible? So I think it is. I mean, we can see the stretch of A and G's. We can go over the possible k-mers, see that there is a specific k-mer that appears more times or appears in each sequence. And it's easy to find it. But what happens when we add 4 mutations for each of these sites? Now when we'll go and look for the k-mers, I'm not sure that we'll discover that this is an enriched k-mer, the stretch of A and stretch of D's, and it's becoming a more difficult problem. So this is an illustration of why it is very difficult. So I'm going to talk about the classic solution, the BEEML algorithm, that uses expectation maximization to solve the problem. And it is the classic problem that given a sequence set, you want to find the PWM that represent the binding sites. So just a short reminder of EM. I'm not going to prove anything about it. So EM, we are giving as input incomplete data originating from a probability distribution, and we have some known parameters. And we want to learn these parameters in a way that maximizes the likelihood. Now, EM helps us in this way to find the parameters that maximize the likelihood, but it does so by finding a local maximum, not necessarily the global maximum. So you may want to start from different positions or initial guesses of the parameters and then maximize and start the EM. And the setting of EM is that we have data X. This is the data that we have in our hands. We can observe it. And we have some hidden information Y that we don't know. If we add Y, we could compute the parameters that give the maximal likelihood. We want to find the parameters. And the main component in EM is the Q function. So this is a reminder, I think, for most of you. Here we have the complete log likelihood of the data. So it's the probability of X and Y, the data that we have and the hidden information, given the parameters. And Q is the expectation over the distribution of Y, the hidden information of the complete log likelihood. And how the algorithm works. So first, we do this iteratively. We first calculate the Q function, and then we want to find the parameters that maximize the Q function. And what EM gives us is a guarantee that with each step, we improve the likelihood. But then we are not sure when to stop. So the most common practice is to stop when the improvement is too small. But it does not mean that we necessarily hit the global maximum. We could hit the local maximum, and we may improve later on much more. But when to stop an EM is a research topic by itself, and I'm not going to cover it here. This is what is usually done for expectation maximization. So what do we do for the problem of finding motifs? So the data that we are giving are all overlapping Elmers in the input sequences. So we can take the sequences and break them into Elmers. And then we assume that each of these Elmers is generated by either model 1 or model 2. So what is the first model? The first model is the Motif model. It is generated by a position weight matrix. So we have A, C, G, and T. And in each position, or each cell in that matrix, we have FIB, which gives us the probability of observing B at position I of the model. So an Elmer can be generated from such a matrix, or it can be generated from a background model. So I'll write that this is model 1. And model 2 is just a vector for ACGT. We have the probability of observing B in the background distribution, in the promoter region, in the region that is outside the binding site. And we also have the parameters lambda 1, lambda 2 that give us the probability that an Elmer is from model 1 or from model 2. So we want to learn these parameters, and lambda 1, lambda 2. And we assume independence between the Elmers, although this is not true. But for simplicity, we can assume that. Now, what is the missing data that we have in this problem? We don't know if an Elmer is generated by model 1 or model 2, by the Motif model or the background model. And if we add this information, we could actually solve the problem very easily. We could just take all of the binding sites, because we know there are binding sites, calculate the frequency in each position, and we are done. So the complete likelihood, in this case, would be the probability of X and Z, given the parameters. Now, since the Xi's, the Elmers, are independent, we can have it as a multiplication of all of those, of Xi and Zi, given the parameters theta and lambda. And now we can use the chain rule and calculate the probability of observing Xi given Zi. So given Zi, we know if it is from model 1 or model 2, so we can calculate the probability of observing that Elmer, and then we have to multiply by the probability of Zi. So if Z it's an indicator variable, so if it is 1, we have this probability, and if the other model, if Zi2 equals 1, then we have this probability. Now when we apply the log on this complete likelihood, then we have it as a sum, and since this is an indicator variable, we can just multiply it by the right probability. So Zij is either 0 or 1, and when it's 1, it multiplies the correct probability. So this is the complete log likelihood of the problem. Now we are going to calculate the expectation of the log likelihood. I'm going to start with some guess of theta and lambda, then repeat until convergence of theta, where we re-estimate z from the current parameters, those of iteration t. Then we're going to maximize and estimate the parameters for the next round from x and z. And then we repeat those steps for various guesses of theta and lambda. Okay, so the expectation that we take is only over Zij because it is an expectation over the distribution of the hidden information. And the only hidden information that we have is in Z, is the indicator variable. That's the only random variable in our case. This is constant, so we can take it out of the expectation. And now we need to calculate the expectation of Zij prime. Let's denote the expectation in Zij prime. Since this is an indicator variable, the expectation is the probability it is equal to 1. And then using the chain rule again, we can have it as the probability of it equal to 1 and of xi, the joint probability, over the probability of xi. And now it splits using the rule of the total probability. So we have the probability that Zij equals 1 and the xi over the possibilities that it is from the first model and the second model. So just to give the intuition quite simply, it's like we have the probability that it is that the Elmer is from the first model, if you want to calculate that it is from the first model, over the probability that it is from the first and the second. So this is the relative weight for being in that model. And we can calculate for the first and the second models. So this is a bit of mathematics. Now we want to maximize it, so we have the expectation. So I'll just give an example of how we find lambda, and I won't go over theta. So in this case, since the log breaks up the multiplication as a sum, we only have to consider, and we only want to maximize, depending on lambda, we can only consider this part of the sum. And so we know that lambda 1 plus lambda 2 equals to 1, so we can replace lambda 2 here by 1 minus lambda 1. Now we just have to derive, right, to find the value that gives us the maximum. We do this mathematics and arithmetics. I'm sure all of you can do that. So we set it to 0, and we find out that the new lambda 1 is going to be the sum of all the Zi1 prime over n. So if you can think about this, is the most intuitive thing to do. We go over the expectation that each Elmer belongs to model 1. We sum over these expectations and divide it by n. So that gives us the new probability that an Elmer is from the first model. And similarly, we can have it for the second model. I mean, if we add z and we didn't have to have the expectation of z, then that's the way we would calculate lambda 1 and lambda 2. So it's very intuitive. And similarly, we can find out how to update theta to the next iteration. And this would be the expected number of times, Cjk is the expected number of times that letter k is produced at column j. So we have that expected number of times over the number of times each one of the letters appears in that position, A, C, G, T. So this is also something that is very intuitive, and that we would do if we had all the data, if we had z. And the way to calculate the expected number of times is again using this z. So once we calculated z already, we can use it to update the parameters. This is quite straightforward, and you can do the mathematics, or maybe you have already seen them in one of the courses that you have taken. So this is one example of to find the one motif, and if we want to find multiple motifs, the common practice is just to mask the one that we find, and then to start the process again. And if we see a big decrease in the likelihood of the model, then it tells us that maybe the next motif that we found is a false motif, but up till then we found true motifs. So just to comment about BEEM, this was on the computational side. It's, I think, the most popular tool that there is out there. And I mean, I just discussed with Steven that one of the wonderful things and features, it has a user-friendly website that biologists can go and plug in their sequences and generate a motif, and it's very fast. They don't have to control the parameters. And in the end, they get these interpretable sequence logos that they can put in their papers. And it's still, I think, the most used motif finding algorithm out there, although there have been other algorithms throughout the years that outperform it. But still, I think at least my impression is that what most biologists need is a nice model that represents the binding preferences. It doesn't necessarily have to be the most accurate, but something that they can later represent and also understand something about the biology behind the process. But it's still an open problem, and there are many computational challenges about motif finding, because many of those motif finding tools find false positives, and we don't always have the way to evaluate the false positive rate and what is true and what is not.
Is it related to the local conversions?
I don't know how much it controls the false positive rate. It might be, yeah. I mean, but usually what you do in this case, you try from different starting points. Yeah, so I don't have an answer how many starting points, how much it has an effect on the false positive rate. And today, MIM is a part of a big MIM suite that has many tools that you can see here. So some are for motif discovery. MIM is here. DREAM, which is for discriminative motif finding. Here it's a problem where we are given a set of sequences which are believed to be bound and a set of sequences which are believed to be unbound. So we have to find a discriminative motif. Other tools, for example, are TomTom to find similarity of a PWM to other PWMs, and there are many other tools. So it's quite a successful suite of tools. Okay, so that was the conclusion of the first part, a reminder of MIM algorithm. So maybe if anyone has a question about MIM, if not, I can go on to the next part. So this will be something completely new, the data and the computational challenges and solutions. So it is time to wake up. And the second part, I'm going to talk about something that was part of my PhD thesis, was the discovering motifs in high-throughput in vitro data. Really something that, technology that transformed the field of gene regulation and the community that goes after these binding models and pushed it forward was the invention or development of protein binding microarrays from Marta Bulick's lab from Harvard. And this was, I think, the first time that we can really get in high throughput and in vitro the intrinsic DNA preferences, binding preferences of a protein. So what is this technology about? We start from a sequence, a design of a sequence that covers each DNA telomer exactly once. And if anyone is familiar with the Bruin sequences, this is how we can get such a sequence. Now we take this long sequence and we chop it up into pieces of 36 base pairs, with overlapping by 9 base pairs, so we don't lose any telomere. And then this gives us the design of the microarray. Now we grow double-stranded DNA in each spot, so each spot on the microarray now has a different sequence, and we know what sequence it has. And using fluorescence A's, we can measure how much DNA we have in each spot. And we can take a protein, which we purify and express beforehand, let it bind the microarray, and use a fluorescence antibody to give us the binding intensity to each of those sequences. So you can imagine what kind of an improvement it is that we don't have any of the factors that involved in vivo binding. Now we have just in vitro a clear signal of what the protein likes to bind. It is to a short sequence of 36 base pairs, not these long promoters regions or ChIP-seq peaks. And it is solely due to a direct transcription factor binding. And what is the data that we see as computational people? We can go to a public database, download one experiment, which gives us the binding intensity of a single protein to many of those sequences. And here, in each experiment, we have around 41,000 probes, each of 36 base pairs, and the corresponding binding intensity. So this is the data that we, the computational people, go and download from the database. These days, a UniProbe database from Marta Bulick's lab, I think, contains more than 400 experiments covering proteins from different model organisms. And also a Team Hughes lab from the University of Toronto as the CIS-BP database that has more than 1,000 of those PBM experiments. So it's really a lot of data. And it's all publicly available. So what is the computational challenge in this case? So we have done one experiment. We've got the data. And if each sequence was the complete binding site, then we were done, right? Because we have the intensity to each of the binding sites, and that's what we want to measure. But the sequences are still a bit long, are 36 base pairs. And usually the binding site is around 8, 10, maybe 15 base pairs. So we need to identify the short binding sites in those probe sequences. And from this data, we need to come up again with a compact model of the binding preferences. This is the computational challenge that we want to tackle. And first, I'll describe one simple algorithm from our lab. And then it was also implemented and used. I mean, it was implemented independently in Team UseLab. And I think it is very simple and still very effective. So first, we are giving the sequences, and we want to give a score for each k-mer. So what score do we give? We just calculate the average binding intensity. So we see TCGA appearing 4 times, and with these intensities, we calculate the average, and this is the score that we give to this k-mer. We do this to each k-mer. Now, here in this illustration, the size of k, the value of k is 4, but what we really do is use k equals 8 or 9, and we cannot do more than that. And the reason is that if you recall, in the design of the microarray, each DNA 10-mer appeared exactly once. So we cannot get an accurate statistics for 10-mers or more than 10-mers. But we can get a good statistics for 9-mers or 8-mers. 8-mers already appear 30 times. So you can have an aggregate statistics over all 8-mers, which is quite robust. So we have this calculation, and the idea behind using the average binding intensity is if it is a real binding site, in all of its occurrences, it will appear with high intensity because the protein likes to bind that binding site, that sequence. But if it is a non-binding site, it may have one appearance with a binding site in the same probe sequence, but in most occurrences, it will probably be with other non-binding sites. So it will have a low binding intensity in most of its occurrences. So we'll get a low average. So this is the idea behind using an average, and it works quite well. And now we are giving this list of k-mers, which gives us a score for each k-mer. Now, if we are interested in a k-mer model that represent the binding preferences, that we are done because we have the k-mers and the scores. But we'd like to now fold it into a more compact position weight matrix model. But we cannot just take this set and fold it, because as you see, there are some shifts in the list. For example, the second one is the TAAT starting from the second position. So we have to do an alignment, right? So we would have to take the second one and move it to the left. We have to have the third k-mer move it one position to the right. Now move it 2 positions here. This one can stay in place, and so on, and we are done. And we can take about 500 or top 1,000 8-mers. Now, in general, multiple sequence alignment is a difficult problem, but this is a very easy problem because the alignment is always to the top 8-mer, which we observed is always the consensus sequence. So the technology is very robust in finding the consensus sequence, and we can align the rest of the k-mers only in reference to the top k-mer. So it's a very simple alignment, no gaps, just 8 base pairs. We can do it very quickly, and then we can look at each position and deduce the probabilities of that column using the scores that we have here. So it's a very simple algorithm. It takes a couple of seconds to run the data and produces accurate models. Now I'm going to move to another algorithm, which is called the BEEML-PBM, and this is considered the state of the art. And the difference here in this model that it is based on a biomechanical model of protein DNA binding. So in the next slides, I'm going to present the model and how it is used in this algorithm. And the parameters of the model are learned using some optimization or minimization procedure. So any interaction, in this case an interaction between a protein and a DNA sequence, can be represented by 2 values, the k on and k off, which are the on rate and off rate of the interaction. So it is the rate that we have these two molecules and they start to bind and we have them bound, or the rate that we have a bound complex and we see it dissolved into the two ingredients. And a way to take into account both K off and Kn is having the ratio, which is termed Kd. So this value represents the absolute binding affinity of a protein to a DNA sequence. And if we have all the Kds, so if for a specific protein we have the Kds for all of the possible DNA chimeras, we can really get the affinities to each DNA sequence. But this is a property of the protein. And these are quite hard to measure, these Kd values, because they depend on the TF concentration, which is hard to measure, and so on. So how is it taken into account in the model? So the probability of binding in equilibrium, so all of these values in equilibrium, and what does it mean in equilibrium? It means that the concentration of bound and unbound DNA is the same. So the probability is the ratio between the bound DNA over the bound and unbound DNA, right? It makes sense because we have the bound divided by the bound and unbound. So this gives the probability for a sequence. Now we can divide both the denominator and numerator by the concentration of the bound DNA, and we get this value. Now if you recall, this is K off over K on times the concentration of the TF, and we can also replace it with Kd. And as I said, we have a problem of measuring this Kd over the concentration of the TF. We have a problem of measuring absolute affinities. We don't know how much protein, in fact, was applied to the microarray and was able to bound the DNA. So what we can measure are specificities. So while affinities give us an ability to compare between different proteins, specificities give us an ability to compare between different sequences, given that they are bound by the same protein. So for a specific protein, we can have the relative binding to different sequences. So now we have this value 1 over 1 plus EI mu, where mu holds the Kd of some reference sequence. So it can be the consensus sequence or some other sequence. This is the concentration of the TF. And EI is the binding model, which I'm going to expand in the next slide. So we need to both learn the parameters of EI and the parameter mu. So what is EI? EI, the energy contribution by the PWM model, can be represented as a PWM. So for example, so here it's a multiplication of the different positions in the PWM model, giving the base pair of SI in position K, and epsilon of that base pair in position K gives us the energy contribution in that position for that base pair. And we can try different models. I mean, we can apply the PWM, we can apply PWM plus dinucleotide features, we can have a Kamer model. So really we can play with the parameters and how we want to represent the energy contribution of the sequences in regard to a reference sequence. Now we should also consider the reverse complement, the double-stranded nature of DNA sequence. So the probability, the binding probability to double-stranded SI is the probability of binding to SI plus the probability of not binding to it, but binding to its reverse complement. So this is modeled. So this is incorporated in this equation. And now, as I said, we have sequences of 36 base pairs. So we go with a window of length L over that sequence and take the probability of each subsequence of length L in that sequence. And this gives us the total probability of a probe sequence. Right, so we have a sequence of 36 base pairs, and let's say we want to, our model is of size 10, so we just sum over all Elmers of size 10 in that sequence, and this is the sum here. Now what is really done in the algorithm is that they take into account some bias in the data, which is called the positional bias. So if you recall, I told you about the microarray technology where we have the probe sequences on the glass. So apparently it is harder for the protein to go into the glass and bind the binding sites which is closer to the glass. It's easier for it to bind the binding site when it is up, not close to the surface. So this is a positional bias inside the probe sequence. So we can add a weight here that represents this positional bias. And they have a heuristic in the algorithm how to calculate the positional bias. I mean, if we look at position 0 to 36, so 0 would be hard, and then the binding intensity improves and goes down a bit. This is how it looks in practice. Okay, so we have the estimates by the model, which is the B of Ti, of the sequence Ti. We have the measured intensities. So now we can fit the parameters to the observed intensities. And then we can apply one of the algorithms to do this minimization. And we also have a term that regularizes the PWM, not to overfit the data or to be too degenerate. So this is an additional term over the position weight matrix model. So this is BEEML-PBM. It's a quite effective algorithm, and it is considered the state of the art. So just one slide on the results and a comparison between the algorithm. I've presented a couple here. I presented rep. This is the sequence logo it generates, and BEEML that represents here. And what we see in general for different proteins is that the sequence logos that rep comes up with, and also seed and wobble, are more interpretable. They have these more strict letters, higher letters if you look at the sequence logo. So this is one advantage of rep over BEEML-PBM, which produces these more degenerate models which are harder to interpret and understand what are the binding preferences. In terms of accuracy, BEEML and rep are on par, on the same level, so they're as accurate. There is one point that I want to mention. In the comparison of the different algorithms, we compared them using PBM data. So we took a model from PBM data and then predicted another experiment from PBM on the same protein. This is in vitro binding prediction. But we also took the model and tried to predict in vivo binding. And we could validate and gauge the performance of how well we do the prediction using in vivo data, ChIP or ChIP-seq. And here you see that the performance decreases significantly from in vitro to in vivo. This is, I think, is still one of the challenges of the community, how to take these in vitro models, I mentioned it before, and apply them to in vivo data. And I think Nadav mentioned a few technologies that give us additional data, such as epigenetic data, nucleosome occupancy, and so on, that we can add and supplement our binding models to improve binding prediction. But I think this is still the goal that the community strives to achieve, and it's still an open challenge. Okay, so where is, yeah.
So you mentioned the positional bias, then how that's accounted for in the BE model. What about in RAP? Do you account for that in RAP as well?
We did not, because we take the average binding intensity, so we kind of assume that if we average over it, it will come out right. So I mean, just by random probability, you have the binding sites appearing in different position. So by the average, you kind of lose this problem. Yeah. This was observed by us and other people as well. Okay, so what is the next technology? As you know, in bioinformatics, we have technologies evolving all the times, and once you have the new technology, the previous one might be obsolete. So I just want to mention the next technology and the computational solutions for it, and some of the open problems in this that are in applying and analyzing the data coming from this technology. So this technology is called the High-throughput SELEX. Have you heard about SELEX or High-throughput SELEX? So SELEX has been around from the 90s, and the thing with SELEX is that you want to measure a specificity. In this case, you want to measure to what sequences does the protein bind. So we can start with a random set of sequences that the protein bind, take out those that were bound, and now take this set of sequences and reiterate the process. So we reiterate the process several cycles, and we get an exponential enrichment of the protein DNA binding specificity. So the exponential enrichment goes into the name SELEX. So SELEX has been in the past with us since the 90s, but in the past we can only do Sanger sequencing. So we were left with a few sequences in the last round that we would sequence and get a few of the binding sites. But now with high-throughput sequencing, we can do the same process, but now we can sequence each round and do high-throughput, not single sequencing, but we can sequence thousands and millions sequences in each round. And what we get in the output, so we do this iteration, this process that we let the protein bind, we take out the bound sequences, we sequence them, and we reiterate the process with the new sequences, we get the sequence files. This is the output of the experiment. So how does it look like in the data? Let's say that in red we have the binding sites. So as we go from cycle to cycle, we see an enrichment of that binding site. And that's what we want to capture, the preference, the DNA binding preference of the protein. And we hope that, the first cycle should be totally random, maybe uniform sequences or close to uniform sequences. And then as we go through the cycle, we see the specificity. So the computational challenge is to take this data and again to come up with a PWM or another binding model. So just to show one advantage of the SELEX technology and why it is so, it's not, we are going to benefit from it compared to PBM data. So the fact that we can generate longer motifs, and this was asked before, that indeed the flanking positions do matter and affect the TF-DNA binding. And in PBM, we are limited by these 8 positions. Right? We cannot do more because each centromer appears exactly once. But here in high-throughput SELEX, the oligos, the DNA sequences that are used, are 40 base pairs. And we sequence them. So we don't have the same length limit. And we can improve the length if we have more sequences, we increase the sample size. So we can generate these longer sequences, longer motifs. And indeed, when we try to use them to predict in vivo binding, we see that they all have better prediction accuracy. So this is just to give motivation on why we are interested in using high-throughput SELEX to improve over PBM. So here I'm going to give and to show one solution.
What was the in vivo binding? Was that ChIP-seq?
Yeah, that was ChIP-seq used. So we took the models that were published for PBM and for high-throughput SELEX for the same protein, and we used ChIP-seq. So first, this is a computational analysis that was part of the SELEX-seq protocol from Bussemacher's lab. And here they first start with finding the K that they want to work on, right? This is a problem by itself. What is the width of the motif of the model that we should use? And the way they do it is using the KL divergence score. So KL divergence gives us the distance between two distributions in bits, or how many random bits we need to add to one distribution to have the other. And they do that over all k-mers that have a count that is more than 100. So some k-mers don't appear very, very few times, so we don't want to use them. But most k-mers appear with a count more than 100. They put them into the calculation where the distribution that they compare is the distribution of k-mers here in the second round compared to the initial round. So we start from a uniform distribution, and close to uniform, and we go to a distribution that gives us the binding preferences that is more specific to the protein binding. And they take all k-mers that have count less than 100 and put them in the same pool, which is legit. So they get a distribution, and they can do this calculation of KL divergence. And where they get the highest KL divergence, they choose that K. So this is sort of a way to measure how much information gain we get from choosing that K in the last round compared to the initial round that we started from. And in their example, it was 12. So that was the choice of K. And then the score that they give for each k-mer is the ratio between the frequency in the ith cycle over the ratio in the initial cycle in a root that is to the I. So like in the power of 1 to the I. And the idea behind that from cycle to cycle, if we see a k-mer, it is multiplied by the same factor from cycle to cycle. So the preference to that k-mer competing with other k-mers is the same factor. And this is the factor that we are trying to figure out here. Now what they really do for the initial cycle is that they don't take the observed frequency, but they estimate them using a fifth order Markov model. Since they claim that they don't have enough sample size in the initial cycle to have good, accurate frequencies observed in the data, they use the fifth order Markov model to generate these frequencies for 12 MERS, which are not that, which not appear with, they don't appear with high numbers in the initial cycle. So this is the score that they get for each k-mer. And now, if you want a k-mer model, you have a list of k-mers and the score. If you want a PWM, you can apply rep algorithm or BEEML algorithm and get a PWM using the same k-mers and scores. So it's quite straightforward. But what is still missing, I think, in high-throughput SELEX analysis is first, how do we choose the cycle? It's unclear if we have several cycles. So far, what everyone has been doing is choosing one of the last cycles with no clear rules, because they give the most specific binding preferences. But maybe we can benefit from looking at previous cycle. No one has been able so far to utilize several cycles together. So maybe by observing what happens through the cycles, we can come up with something more accurate about the binding preferences than just looking at a single cycle. And of course, with every technology, we have technological biases, so we need to cover them. And as I mentioned before, this is one of the big challenges that the community of gene regulation is still trying to solve. So this, just to summarize these two parts that I've discussed so far. So I've shown different models that we can use to model DNA binding preferences, where the PWM is currently the most popular and used. I've done a reminder for the MIM algorithm, how to use expectation maximization to find motifs, and also talked about the new data from PBM and high-throughput SELEX, and about the computational analysis to perform on this data to get the same motifs. So this concludes the second part, and I'm going to go into other problems. But if someone has a question about this stuff.
Could you comment a bit on the meaning of the weights, either in the PWM or in the BEEML model?
Yeah.
Because sometimes it's a probability, sometimes it's a regression to predict the...
Right. So in the BEEML model, it's the energy contribution. It goes in that model that I've shown. So they say it's the energy contribution of that nucleotide in that position. Where in other cases, it's the frequency. So these are like common weights. Another way is to have a log odds ratio in each cell. So you have the log, and then it's a weight that you sum over. So you have the frequency of the base B at position I. You go over the frequency of that base, let's say in the background, and then you take the log. So that is the log odds ratio. So how surprising it is to see the base giving the background distribution. And then you can sum up the logs and get a score for the binding sites. So these are common weights in the matrix. I hope that I answered.
Yeah, for example, in the RAP model, you showed weights which were frequencies of the top k-mers. So
Yeah.
it seems like it's a probability. That the k-mers were selected because they had a high RBM value. So I was wondering whether you had some implicit assumption, like high probability means...
Yeah, so I don't have, we just folded it into a distribution. So the sum in each column would be 1, but it doesn't have to be necessarily so. So it's not a frequency, right? It's a binding intensity. Yeah. So yeah, it's a mixture of binding intensity folded into frequency. So yeah, there is something about it that we didn't think about. Yeah.
I have a question about HT SELEX. Do people do replicates with the same transcription factor, and how similar are the actual sequences that protocol converges to?
Well, you don't care about the sequences but about k-mer frequencies at the end.
Sure.
Because you might start with a different pool of oligos. It's a random pool. So no one has looked at the exact oligos, the 40 base pairs long oligos, but they did look at the k-mers. So we in our paper compared, so they did replicates both for, and we didn't compare replicates, but we compared mouse and human proteins and full proteins compared to only the binding domains. and we didn't see any significant difference between those, but we only analyzed the K-MER frequencies and not the complete oligos. So, are you looking for something in the... yeah. So in that comparison, we didn't see any significant difference.
We had a paper in the Annals of Applied Statistics where we actually tried to look at several stages simultaneously. You might be interested.
Yeah. So I can catch you up later. Yeah. Okay. So I'll go to the slides which I kept as a backup. And this is a new theoretical idea how to use the Bruin sequences for sequence design and assembly. And this is bidirected Bruin graphs. Okay, so does anyone know about bidirected graphs? Okay, so I'll be showing something new to the audience. Just about the motivation for the problem. So assembly, we all know what assembly is, right? We heard from Lior and Pavel Pevsner. And the sequence designs come into play in the PBM technology, right? I mentioned that they have a design where each 10-mer appears exactly once. They use the Bruin sequence. So this is where these kind of problems come into. Okay, so let's start with the introduction. I think most of you, all of you know what is the Bruin sequence of order K. It is a cyclic sequence that every possible K-mer appears exactly once. This is the sequence that they used for the PBM technology. It is the most compact sequence. Well, indeed, each K-mer appears exactly once. And so the length is the size of sigma, the alphabet, to the K. In our case, it's the 4 nucleotides, so it's 4 to the K. And here is an example of the Bruin sequence of order 2, right? If you want to check me, you can go over the sequence and see that, indeed, every possible dinucleotide among the 16 dinucleotides appears in this sequence. How do we generate the Bruin sequence? So one way to do it is using the Bruin graphs. And here is an example of the Bruin graph of order 1. And what we have in the graph is every vertex has a label of a K-mer. So we have the size of sigma to the K K-mers. Every edge, a directed edge, is an overlap, as Lior mentioned in his talk. So it means that we have an overlap of K-1 between the source and the target vertex. So the number of edges is the size of sigma to the K+1. And we get that the in degree and the out degree are equal, and both are equal to the size of the alphabet. As a fact, we get that the graph is strongly connected and each vertex is balanced. And if you recall from Algorithms 101, this means that the graph is Eulerian, and we can perform an Euler tour over the graph. It means that it covers, this Euler tour gives us the Bruin sequence of order K+1. So this is one way to generate in linear time the Bruin sequence. Here in this example, you see the Bruin graph of order 1. Each nucleotide is represented as a node, and all possible dinucleotides are edges in the graph. Okay. So other ways to generate, just as a comment, the Bruin sequences are linear feedback shift registers, if anyone heard about them, and there are recursive ways to generate those, but this is a very useful way. Okay, so how do we do assembly by the Bruin graphs? And I mean, we look at the reads. We have the set of K-mers that appears in the reads. We can construct the Bruin graph and have the edges represent the K-mers. And then we can perform an Euler tour and find the sequence that connects all of these K-mers together. Now, is it always possible? So not necessarily. It depends on the set of K-mers that we have in the data. And what we have to do, in case it is not strongly connected, or of course if it's not strongly connected, then we have to work on each strongly connected component independently. But if not all of the vertices are balanced, then we have to cover some of the K-mers or the edges several times in order to go and cover all K-mers. So our aim is to have a sequence, one sequence that covers all the K-mers that we see in the data. So we can solve it using the Chinese postman, the solution to the Chinese postman problem. I didn't know why it was called the Chinese postman problem, But one Chinese student said that it's a big problem that they learn in China, and it's because Chinese mathematician studied and gave the solution. I don't know if this is the case. And what is the problem of the postman? It has to go over each road and deliver the mail, right? And it is something. It has to go over each road, so each road or street is represented as an edge in the graph, and it has to go over and deliver the mail. It may go over a road several times. So find the shortest path that traverses each edge at least once. So we may have weights on the edges. They can represent, if it is like k-mers from the data, the coverage of the k-mer or the likelihood of that k-mer. So we have weights and we want the shortest path, and it can be solved by minimum cost flow. So we can define as a set V minus all the vertices with greater in degree, and with V plus all the vertices with greater out degree. Then we can have the capacity from the source to all of the V minus vertices be the imbalance. So this is the number of edges that we'll need to go over or to add to have them balanced. And similarly from the vertices in V plus to the target. And all of the rest of the original edges can have a capacity of that is unbounded. We can go over them as much as we want. What is the cost for the different edges? So those that go from the source or to the target can have a cost of 0. We know how much flow is going to pass through them, but all the rest is the original weight that is part of the problem. I mean, the cost of traversing a street or the likelihood of a k-mer or so on. And then after we found the minimum cost flow, what we do is we can add for each edge UV the number of copies of that edge according to the flow that passes through that edge. So that fixes the balance problem for each vertex. So this is the Chinese postman, and there is some about how we can solve the problem of minimum cost flow using successive shortest path. And I give you several bounds about how to find the shortest path and so on. Now, so far I haven't described anything new because we know how to do assembly. We know that most of the assembly algorithms work in this way. How can we improve? So we have to consider the reverse complement, the double-stranded property of DNA, which means that if we have a k-mer, we also have the reverse complement of the k-mer. So in this example, if we have this sequence, we also observe this sequence. And as I say here, in a double-stranded sequence, when we have the k-mer, we have its reverse complement. So the goal is to find the shortest sequence to cover all the k-mers. Now a k-mer can appear as itself or as its reverse complement. So it doesn't necessarily we have to cover the k-mers as they are in data. So maybe we can find an even shorter double-stranded DNA sequence that covers all the k-mers that we observed in the data. And one way to do it is using bidirected de Bruijn graphs, which I'm going to introduce soon. And I'm going to give a solution to the problem using linear programming and also using a bipartite graph. And I'll also describe an application to the design of the PBM probe sequences. So what is a bidirected graph? Maybe I should have this erased. Room to write some of the definitions. Yes, this is a new notion to some of you. So here, each edge endpoint has an orientation. Okay, so here, if we have in the example, we say that we have x, and then this kind of orientation is a negative orientation, and the orientation of edge b in respect to x is positive. Now each edge point has this orientation, and the incidence matrix values of -2, integer values from -2 to 2, which is for each edge. If it has 2 negative endpoints, it is -2. If it's 2 positive, it has 2, and can be anything in between. Now the in degree is the sum of all of the edges that have a negative endpoint, and this is all of the edges that have a positive. And the balance is the out degree minus the in degree, as we know, and also the sum over the incidence matrix. This gives us the balance of each node. Okay, so this is bidirected graphs. How does it relate to double-stranded DNA? So just before I go on to that, to the De Bruijn graphs and how we use that, so a walk now is not the same as a path in a directed graph. Now a walk has to follow these rules. So it is still a series of vertices interleaved by edges, where EI is incident to xi and xi plus 1. But the edge has to follow the rule that EI minus 1 and EI have opposite orientation at xi. So this is an example of a walk. So we have W and then A and X. Then we go out of B. So here A entered with a negative orientation and we went out of X on B with a positive orientation. So this follows the rules, and this is considered a walk in a bidirected graph. This, for example, is not a walk. So we go to A, then to X, and to this. So this is negative, and this is negative orientation. So this is not a walk. So we have to remember that we don't have paths, as we know from directed graphs, but we have walks. So what are bidirected De Bruijn graphs? So in the original De Bruijn graph, each node here represented a k-mer. Now each node is going to represent a k-mer and its reverse complement. So that's the addition, and that's why it's so useful when we want to do assembly and consider both strands, also for sequence design. And there are some rules about what is the label of this edge. So for example, if E is positive to X, so we have here that it is positive here, and negative Y. No, so this is positive to both of them. E is positive to X and positive to Y. Then Px, AT, overlaps NY, which is TT. Okay, so this rule worked. We have, so the label here would be ATTT, ATT, and right, so I won't write on the wall, don't worry. This is going to be ATT, and the reverse complement is AAT. So this is the label of this edge. Okay, so now each node as well as each edge represents a k-mer and its reverse complement. So how can we solve the Chinese postman problem? So again, we take the k-mers that we saw in the data, we put them on the edges, we put each k-mer as an edge in the graph. And now what we have to solve is the minimum cost flow, and we can do this using linear programming. So we want to minimize the number of flow that we put times the cost of traversing a flow through edge E. And this is subject such that the flow that we add fixes the balance, right? We had the imbalance in each node that we wanted to fix, so we have an Euler tool in the graph. So we can have this constraint that the flow that it now passes through the edges has to fix the imbalance for each one of the vertices. And of course, the flow has to be non-negative. And we can run this and get a solution to the problem and get the shortest double-stranded DNA sequence that covers all k-mers. So here are some technical details about the LP and the fact that we are getting it to approximation using this notion. And in fact, we can do it in an optimal way and polynomial time if we look at the following bidirected graph. So let's do it here on the board. We add a set of sequences that add a imbalance that add a greater in degree. We add a set of vertices that add a greater out degree, and we need to fix the balance. And the only way we can fix this balance is by adding a path from a node here to a node here, right? And then we add an edge here, we add an edge here, so we fix the imbalance for each of those by +1. I mean, so now the imbalance is reduced by 1 for each of those. So we need to find these shortest paths. Then all we need to do is a bipartite matching between these vertices and these vertices, or the imbalance of these and the imbalance of these. Then we can solve the problem in polynomial time, given that we have a solution for the bipartite matching problem, for the weighted bipartite matching. So this is in fact what's written here, and the only thing that we need to consider is that we are talking about bidirected graph. So now finding the shortest path is slightly different, but in fact it's almost the same algorithm, that instead of looking for example for the Dijkstra algorithm that finds the shortest path, instead of looking at the shortest of any neighbor that we can go in the next edge, we have to consider neighbors that are consistent with a walk. As I told you, now we don't have paths, we have these walks. So we can compute the shortest path between these vertices and these ones, and then we can solve it using a bipartite matching. And this gives us an optimal solution in polynomial time. And then once we have added these edges by the flow, we can find an Euler tool and find the shortest double-stranded DNA sequence that covers all these chimeras. Okay, so here it's a bit about the running time. I'll go over it. But, these are common algorithms that we know from graph algorithms on running polynomial time. And now it is related to the sequence design problem for PBM, right? So this was, I talked about gene regulation. And in the PBM technology, we have a coverage of each DNA telomer exactly once. So in fact, we can reduce the number of probe sequences by having a coverage that for each DNA telomer either covers the DNA telomer or its reverse complement. Does not need to cover both of them. So that way we can have a reduction by half of the number of probes that we can use in the technology. And maybe we can increase it to k equals 11 instead of 10, now that we have more space on the array, or we can add more sequences of something that we want to measure. So there is a high incentive to have a more compact and efficient design. So for odd k, the graph, the bidirected De Bruijn graph, is balanced. Every node in that graph is balanced. And here is an example of one node and the edges that are incident to it. But for even k, there are a few nodes that are unbalanced. The problem comes from palindromes. This is something that Lior asked about yesterday. What is the problem with even k? Why don't we use that? We have the palindromes, the reverse complement palindromes. So here, ACGT is the reverse complement of itself. And for that reason, if you build a graph, it will have a positive orientation at both of its endpoints, and now this node is no longer balanced. Okay, so now we want to apply the same algorithm as we just did for the assembly problem to fix the imbalance of the nodes in this graph. So we can apply the same polynomial time algorithm and then solve the problem. We in fact can do it in a more efficient running time because all of the weights are integers, and we can improve the running time of Dijkstra and the bipartite matching. So just to show you the computational results, so comparing between a De Bruijn sequence and a reverse complement De Bruijn sequence. So I don't show any results for odd k because for these cases the length was just reduced by half. But for the even k cases where we had to do this fix because of palindromes, we see that we have a reduction that is almost by half. It's approaching half as k increases. Here we have the numbers. So in practice, we are able now to give a design that reduces the number of probes by half, both for even k and odd k. And this is an optimal solution for both k's, for any value of k. Okay, so some open questions. So there is also some interest, and that was done in the original design of PBM technology, to cover gapped k-mers. So can we improve that? Because here I only considered contiguous k-mers. What is the number of reverse complement De Bruijn sequence? So here, this is the equation for all possible De Bruijn sequences. And just for theoretical interest, I mean, I don't think it has any application or relevance to the technology or biology, but we can ask the same question. Can we have a design that minimizes the number of probes that cover the set of k-mers? Because in the solution that I introduced, we find one Euler tool that covers all k-mers together. So this is one sequence that we then have to chop up and use as probes. But since the technology already uses the set of probes, can we design the probes directly without going through one long sequence? And can we suggest a linear time algorithm because the polynomial had some, like it was, I think, 4 to the 3k over 2. Maybe we can improve that to the 4 to the k. And then can we come up with a closed formula to the length, because all of these values are from empirical tests. I mean, we ran the algorithm and we got a result. Okay, so I think this is a reference for the last part, but this concludes the talk. So I've got to talk about 3 parts, about the motif finding and about the sequence design problems. Yeah, so I'll be happy to take questions, and thanks for listening.
Any questions?